My name's Parker Moss. I'm the Chief Commercial Officer of Genomics England and one of the leaders of our research community as well. It's such a pleasure to be here on the NVIDIA stage. NVIDIA is a company I've admired for many years, not just technically, but also in your remarkable ability to have terrific timing on impacting industries such as ours in life science. And I hope in my presentation today I'll explain how ideal the timing of bringing GPUs at scale into the life sciences industry has been. So what I'd like to talk to you about today is a little bit about Genomics England itself. Then I'll talk about how machine learning is used in genomics today, and I'll give you a few case studies. And then I'll move on to the kind of infrastructure that we require at Genomics England to drive machine learning in clinical diagnostics and research at scale nationally with the NHS. So Genomics England is a very unusual company. We are a private company, but wholly owned by the Department of Health, and we were really conceived by the ex-Prime Minister David Cameron as part of the Olympic legacy for the UK. So David, as many of you will know, had a child with a rare disease, Ivan, who very sadly succumbed to that disease. And like many parents of children with rare disease, he developed expertise in that field, and he realized that the future of rare disease and indeed cancer research would lie in the whole genome sequence. So in around 2013, David Cameron created a moonshot ambition for the UK to be the first country in the world to achieve 100,000 clinical genomes sequenced in cancer and rare disease cohorts. This was considered impossible by many. At that time, genomes were expensive, the logistics were unclear, and it was a very challenging bioinformatics and pathological process to make this happen in a clinical context. But Genomics England was formed really as a national science project to make that happen, and we worked towards that between 2012 and 2017. And by the end of the 100,000 Genome Project, we had successfully sequenced actually about 120,000 whole genomes from patients in the NHS. Now, while we will continue to support and serve those participants for the rest of their lives, we've finished curating that data set, and we've taken time to sit back, pause, and reimagine our vision for the future. And I think we've come up with something important and impactful. The 100,000 Genome Project itself generated an enormous amount of data. So as I mentioned, we sequenced 100,000 genomes from 97,000 participants. Now, of that were cancer genomes, 37,000 tumor normal pairs. So we sequenced both the germline that you inherit from your parents, but also the somatic sequence, the sequence removed from a surgical biopsy of a cancer tumor. In rare disease, we sequenced 74,000 rare disease patients, and for over 80% of them, we got at least one family member as well, and that's extremely important for understanding the heritable elements of those diseases. We generated 54 petabytes of data, which today sit in our Amazon Web Services instance. And of course, genomic data is no use in itself without clinical data. And so thanks to deep partnerships with NHS Digital and Public Health England, we have extensive retrospective and lifetime follow-up on all of the patients in our data set, including hospital episode data. We have the status on their mortality from the Office of National Statistics. We have full-text pathology, full-text radiology, full cancer regimen, longitudinal data, and it really creates an enormously rich data set for research and for clinical interpretation. So we've established a simple but impactful vision, which is that everyone should benefit from genomic healthcare, and everyone is a really key word in that sentence. And to instantiate that vision, we've built a strategy which has two parts, a healthcare part and a research part, and I'm going to just talk you through both of those right now. So in healthcare, as you would expect, it starts with the patient. And the first thing that we've developed with our patients is the ability to put them through an extensive consent process. Everything we do with our participants is highly consented, and we consent participants to have their genome sequenced, to follow up their life course clinical data for the rest of their lives, and also to get access to all of their retrospective data. We consent them to allow us to re-engage with those participants so that we can re-phenotype or recruit or even resample those patients, and those samples can be vital parts of functional genomic studies. And we consent participants to allow research to take place on their data, de-identified of course, and that's both by academic and biopharma partners of ours. So having completed consent, we sample those patients in clinic. Those samples travel to our sequencing center, which is in Cambridge and run by Illumina. The outputs of the sequences go through our custom bioinformatic pipelines, which are ISO accredited, clinically accredited pipelines, which we write in-house. And eventually the outputs of those pipelines, which are lists of highly characterized and well annotated pathogenic variants, go back to 7 genomic lab hubs, which are run by the NHS, which process those lists of variants into diagnoses, and that gets back to the patient in clinic and affects their care pathway. So it's a very extensive and comprehensive process which generates a lot of data. Now that data, both retrospective data from the 100,000 Genome Project and the prospective data that we're collecting, travels into our research sphere. And our research strategy is really to make sure that the best researchers from around the world have access to comprehensive genomic data from our participants. So we have the ability to first accredit researchers and then instrument them with bioinformatic tools and then give them access to our data. And we've recently relaunched our bioinformatic platform, which is a wonderful company called LifeBit that provides really intuitive and immersive bioinformatic tools, all on the data which sits within our Amazon Web Services instance. And now for the last part of our data architecture, which is the return path back into our clinic. One of the things that makes us really unusual as an organization is that all of our data is captured from participants identifiably. So whilst we only make de-identified data available to researchers, in-house we can re-identify patients. Now why is that important? Well, on a regular basis, researchers in academia and also in industry make discoveries about patients' diagnoses, discoveries that have potential therapeutic implications about new gene therapies or cell therapies that could be applicable to those patients, in particular young children who are suffering from rare disease and with intervention therapeutically could have those diseases reversed. And so we have the ability to review those findings to make sure that they're ethical and accurate, and then we can re-identify the patient and pass those results back to the patient's clinician. So in other words, our operating model is inherently translational, and we have a constant cadence of diagnostic discoveries and also changes to treatment pathways that go back to our participants back in the NHS. And that makes Genomics England an incredibly compelling and an emotionally rewarding place to work within. Now, of course, we can't do this on our own. This requires us to work with the entire UK ecosystem, and we're deeply connected to the NHS, but also the other organisations in the UK that make our health system work. So we have David Pryor, the chairman of the NHS, on our board. We have Sue Hill, who's the chief scientist of the UK, who observes our board and is in daily contact with us. And everything we do is in close coordination with the main bodies in the UK, including Cancer Research UK and HDR UK and the National Institute for Healthcare Research, which we work closely with. We also work with 4,000 academics, about 20% of whom are regularly in our environment and engaged deeply in our data. And with our commercial partnerships, 8 of the top 10 pharma companies engage and use our data in drug discovery and in clinical development for their clinical trial platforms today. So I've discussed a bit about the data in the 100,000 Genome Project, the retrospective data, but we're adding new data every day, and that comes through the NHS Genomic Medicine Service. So since about November last year, we have been really acting upon our strategy to impact healthcare in the NHS by whole genome sequencing both rare disease and cancer patients in the NHS. And this is a really exciting world first. In rare disease, we've been sequencing 30 indications of rare and ultra-rare diseases. And in cancer, we started out with acute leukemias. With pediatric cancers, we sequence all pediatric cancers at frontline, but also upon relapse, and sarcomas. And I'm delighted to say that we've just had approved to extend our cancer sequencing to include ovarian cancer, that's high-grade serous, triple-negative breast cancer, which is the most severe subgroup of breast cancers, all of the brain cancers, other hematological cancers like multiple myeloma, and we've got a bunch of pan-tumor relapse and refractory conditions that we're also sequencing. So this is really exciting. It's about 12 to 14% of all diagnoses will get a whole genome sequence, which will be funded and then returned to the NHS, and this is an absolute world first. There are some really gold standard hospitals in America that are doing whole genome sequencing for some of their indications in some of their centres, but we're by far the first country in the world to be doing this at a national scale, throughout the NHS, providing whole genome sequence analysis with complete equity of access across the country. It's really a remarkable achievement and commitment of the NHS to go this way. We've got some new initiatives as well, and many of them are technologically fascinating. So in rare disease, we are digging deeper into the rare disease patients who we've not managed to find diagnoses for, and we're adding transcriptomics and low and high abundance proteomics to their analyses as well. And we've been doing that with some research partners around the world, including Illumina. And we really hope to increase the diagnostic yield of those rare disease patients. And then the natural extension of our rare disease program is a highly exciting area called newborn sequencing. So Genomics England intend to encourage the UK to be the first country in the world to whole genome sequence all newborn babies at birth. And we'd like to do that by kicking off a trial with 200,000 babies that we are aiming to launch next year with support of the NHS. And this is something we've been working through for many years now. We've been conducting extensive public dialogues and ethics projects, and it looks like the public are prepared to support this very bold platform, which will be both a diagnostic and a therapeutic platform, and we think could identify young children early in their life course that would still be susceptible to reversals in their disease progression with some of the new and exciting therapies that are coming online, like antisense oligonucleotide therapy and some of the RNA therapies that are coming out of the cell and gene therapy community. So that's an absolutely thrilling program. In cancer, we're exploring a new type of sequencing called nanopore sequencing, which is really being led by this terrific company, Oxford Nanopore, a UK champion that's just IPO'd, and we're doing that sequencing in cancer to look for new structural variants and to look for methylation, which I'll talk about a little bit later. And we also have a radiogenomic program in cancer in which we're going to be adding radiology and digital pathology images to our genomic data, which will allow us to look at both the molecular and also the spatial context of tumors. And moving away from the technology, another big ambition of ours is to increase the ancestral and socioeconomic diversity of our data set, and we're investing heavily this year to make sure that our data set is truly representative and that we're recruiting from all communities across the UK. So these are some of the exciting new initiatives that we're working on. So let's talk about machine learning. This is a machine learning event, and I'd love to tell you how the AI/ML world is contributing and really rocking the world of genomics. So to start with, we'll have a little bit of a talk about how computers learn, and I recognize here that we have some real experts in the audience, so I'm going to go fast here. In principle, there are three major modalities of machine learning, and I'll go through them in order of simplicity to complexity. So we start with supervised learning, and supervised learning in healthcare is most commonly associated with image classification for diagnostic analysis. So on the screen here you can see what is actually a very, very clear collarbone break. It doesn't require a radiologist to see that. And the arrows are a simple cartoon of an annotation that points out the features of that break. And what supervised learning does through neural networks is to train a network to review thousands of images of similarly annotated features, and by the time it's trained, it will recognize on its own a subsequent 1001st or 1002nd image and accurately identify features like collarbone breaks. Reinforcement learning gets a little bit more complicated. So here's a beautiful example of reinforcement learning, which is well known in the machine learning field. It's of a classic Atari game, and in this case the neural network knew nothing about the rules of the game. It wasn't told what a paddle or a ball was. It was just simply told to manipulate the parameters available to it and to maximize the reward, which is represented in the score at the top. And what was really astonishing was that not only did the neural network quickly learn how to play the game, but it also learned how to master the game. Look how it's sending the ball up the left-hand side, building a channel so that the ball goes above those blocks and eats them from the top down. It essentially achieved mastery status of this Atari game in an impressively low number of iterations. Now, the interesting thing about supervised and reinforcement learning, for those of you who have children, is that this is very much how we teach our kids. So we take a young child onto the street, We show them cats and dogs and help them differentiate between them. We show them cars, we show them dangerous roads, and we hope that after some time, children will recognize the features in real life which are dangerous and to be avoided, and that after some time, they can go into the world unsupervised. They can identify features on their own without previous training, and that they can become adults. And essentially, unsupervised learning is really where things get exciting in the life sciences world. So before talking about unsupervised learning, I first wanted to draw your attention to a disruption which I think we should all be aware of, and which is really quite remarkable. So many people in this audience will be aware of disruptions like, for example, Google really commoditizing the neural network by making TensorFlow available, or indeed NVIDIA itself commoditizing parallel processing by bringing GPUs into the mainstream. And in my field of genomics, what is remarkable is that in the last 20 years, a whole genome sequence has gone from $1 billion in several years of analysis down to a few $100 and a few hours of work in the wet lab. So these are remarkable disruptions, and they are the kind of disruptions that we have learned to expect happen every 10 to 20 years. But there's been another disruption which I think is really a once in an epoch thing, and it's a disruption to the scientific method. And that's why I have these great famous scientists who are masters of the scientific method up on the screen now. Let's take Newton as an example. So Newton had strong views about metaphysical laws that he felt would describe the world. He had a language to describe those laws in. In his case, it was geometry and trigonometry and calculus. And he had a huge brain upon his shoulder in which he could hypothesize how experiments could be built that would test his view of the world with inputs and see whether they could measure outputs reproducibly and consistently. So this was really a hypothesis-led way of conducting science. It's the central scientific method that has led to where we are now over thousands of years. It involves creating inputs like apples falling from trees or observing billiard balls traveling on billiard tables, testing that against the hypothesis, measuring the outputs, and if the outputs indeed correspond with what we've hypothesized, then a scientific theory becomes law. Now that works extremely well with simple systems like here the Newton's cradle, which is involved with measuring the position and the momentum of 5 balls on the screen. And ultimately it's a reductionist process. It's a process of reducing a network down to its simple principal component parts and analyzing and then predicting their outcomes. But in biology, networks get much more complicated than that. Look at this Circos plot, which describes the somatic sequence of a cancer genome. The ring around the edge are the 22 chromosomes which are common to all humans, and then the X and Y chromosome of the male and the female. And the lines in the middle represent, and this is not a cartoon, this is a real representation of mass-scale translocations, fusions, and rearrangements of genes that have been dysregulated by the oncogenesis process. So this is what cancer does to your genome. So this is a cell which has been analyzed going through the process of cancer. And that just sits within a wider network. So this is a remarkable image that I wanted to share with you all that Roche has produced, and it's publicly available. And in the center of the image is the citrate cycle, which shows how fat is metabolized by the body. But if I let the cartoon run, you will see that we can pan out and you will see all of the other biomedical pathways that this image interacts with, and it's absolutely enormous. Everything you see on the screen here is a different input and a different output. And I think it's clear from here that you can see that even if we did have the biological language to describe the inputs and the outputs of this system, and even with the enormous brains of the scientists today, it is very unlikely with a system of this complexity where all of the metabolisms are linked with each other that we would be able to lead with a hypothesis and then test the impacts of inputs upon outputs. We need a new tool in our armory, and machine learning came along at just the right time. So here's the new method of doing science. Scientists measure inputs, we measure outputs, we run that through neural networks, and the machine itself hypothesis generates. It, in a sense, turns the scientific method on its head. So in a sense, we've moved from reductionism to radical empiricism, which is a really great term that explains not just that we learn from the evidence we observe, but we have to do so radically because we're taking in thousands and thousands of inputs. And this was a term that was first proposed by William James, a 19th century philosopher, but has been picked up upon by Zavain Dar, a great thinker and professor and also investor in California, who applied radical empiricism to the application of neural networks in many fields today, but I think it is particularly relevant to life sciences. And I just want to pause here for some of the scientists in the room, because some of the scientists may feel that what I'm actually saying is that we have eliminated the use for scientists today because we're essentially hypothesis generating through a machine. And that is absolutely not what I'm saying at all. And let me just explain that. Perhaps we could say that there's no need for chess players or Go players or checkers players today, because ultimately the game of chess has been solved. And other than doing it for entertainment, there's really little to be gained by humans playing in chess today. But that is not true of science. And what is the difference? Well, we wrote the rules for chess. We wrote the rules for Go, but we did not write the rules for biology. We were makers of chess, but we're takers of biology. And the best that these machine learning networks can come up with are well-characterized associations between inputs and outputs, but it still takes great and insightful scientists to really look at those associations and determine and draw analytical meaning from that. So scientists are still very, very much needed in this realm. So with that background, let's see how all of that applies to biology itself. So this is a beautiful image that I've put in front of you, but it's a rather dark image itself, because this is taken from the surgical biopsy of a very unwell patient. This patient is suffering from mesothelioma, which is a cancer of the pleura of the lung, likely gained through asbestos exposure, and very sadly, nearly all patients with mesothelioma die. And this in itself is a complex system. Now, how do we analyze complex systems? Well, many of you know that dogs and cats were central to the way that we have learned and taught machine learning networks to recognize shapes, colors, textures from images that were uploaded on the internet. And thanks to transfer learning, we can apply those shapes and edges to a complex digital pathology image like this. So this is really taken from a case study by a very interesting company that I used to work with called Alkin, who was set the challenge of understanding the prognosis of patients with mesothelioma. Now this image is pink and purple, as you can see. It's a very standard image in my realm. It's pink and purple because it's been stained with hematoxylin and eosin, and the dark purple binds to the cancer cells, and the lighter purple shows the stroma surrounding the cancer cells, and there's even some white which shows other features of the microenvironment. Every cancer patient has their disease analyzed this way. Now, what the data scientists in this program were able to do was to collect 10,000 digital pathology images. These are very high resolution images from mesothelioma patients, and they actually broke those images into 10,000 tiles themselves. So we actually had about 100 million small images, small tiles, to train a neural network from. Now, those images were pre-processed into the 100,000 tiles. There was some feature embedding to reduce the complexity of the image itself, and in fact they were turned into black and white. They were put through sorting layers, and ultimately they were put through a multi-layer neural network, which just simply classified each tile into cancer and non-cancer. And then those tiles were mapped back onto the master image of the tumor itself. And what you can see, which is really interesting, is that amongst these patients, two subgroups emerge. Some patients survived on average for 32 months, some patients survived for about 8 months. This is something that clinicians had recognized, but clinicians and pathologists who had stared at these images for many years had never been able to explain or to prognose. But the fascinating thing is when you look at the tiles that are associated with high and low survival. And even from the untrained eye, you can see from this page that there's a textural difference between those images. And when we map those images back onto the master slide, you can actually see just about here that the high survival tiles cluster slightly around the edge of the image and where the microenvironment is, and the low survival tiles are much more associated with the cancer cells themselves. Now, these features of the digital pathology image were something that pathologists had been aware of for many years, but they were unaware of the prognostic implication of this. And this became hugely exciting. It turns out that for decades, pathologists had been staring at the wrong thing. They had been looking at cancer cells rather than the stroma, and in particular the vascularity of the stroma in the microenvironment of the tumour. And it was actually the non-tumour cells that were driving high or low survival outcomes for these patients, which is an astonishing morphological observation, really driven by an unsupervised, or at most a very weakly supervised analysis of these completely unlabeled digital pathology images. So what had we come up with? We had come up with a way of prognosing patients from unlabeled images, but we also came up with new biology which could explain the mechanism of action of this very, very difficult disease. And this can lead on to exciting drug discovery pathways. We can stain the stroma in other ways to look for proteins that are associated with high survival and low survival. Perhaps we can develop drugs to downregulate or disable these low survival proteins, and that is essentially the essence of a drug discovery program. So let's move from a pathology example to the business of Genomics England, which is the genomic code itself. Itself a very complex system of A's and C's, G's and T's. And as I described before, in cancer and other rare diseases, these bases can be radically rearranged by disease processes. And it doesn't stop there. DNA gets transcribed into RNA, which has different levels of gene expression, and then an RNA makes proteins which we've seen have been incredibly predicted with DeepMind's AlphaFold, where just from the two-dimensional amino acid string from DNA, AlphaFold have essentially now solved the problem of predicting structure from proteins, something that I think none of us felt was possible only a couple of years ago. And I'll give you a real example of some machine learning analysis that has been done on Genomics England DNA. So this is a remarkable lady called Serena Nick-Zainal. She's a researcher at Cambridge. She recently won the Steiner Award for Cancer, which is a bit like winning the Field Medal in maths, and she's been working on breast cancer images. So these are 3 circus plots which were created using Genomics England data. And if I zoom in the middle, you can see something quite remarkable about these plots. So to the untrained eye, this won't be obvious, but after we apply an algorithm to these circos plots, the algorithm can pick out features of pathogenicity. The image on the left is of a lady who has ER-positive breast cancer and is likely to have a good outcome from standard of care treatment. But the genomes represented by these circus plots on the right have more challenging outcomes. And what Serena has done is she's applied actually an AI statistical approach. She used logistic regression, a lasso approach, to look for features in these genomes which could explain the outcomes. And she discovered a few remarkable things. So from the image in the middle, she identified a type of DNA damage repair called mismatch repair. And mismatch repair goes on to generate microsatellite instability, and the importance of that feature is that there's a new class of drug called an immune checkpoint inhibitor which binds to MSI and significantly improves the outcomes of patients that have access to that drug if they exhibit that very precise marker of genomic instability. So that was a tremendously important finding for that patient. And the one on the right is even more important. So it has been known for some time that another marker of genomic instability, which is called homologous recombination deficiency, is very common with women who have BRCA mutations, BRCA1 or BRCA2 mutations. Unfortunately, there's only about 5% of the women, and I say unfortunately because women with this HRD markers respond very well to another class of drug called PARP inhibitors. And what Serena discovered by studying the genomes in the Genomics England dataset was that actually BRCA-ness is a feature which is also presented in the non-coding region of the genome, and which looks like it may actually apply to more like 15 to 20% of women. So it could be that through this analysis, and this is all to be proven out through clinical trials, we could find that responders to PARP are 3 to 4 times as prevalent as we thought they were originally. So this could save tens and tens of thousands of life years for women in Britain and ultimately around the world. An incredibly important observation. So that's how we analyze individual patients. but Serena's analysis, alongside our own internal researchers at Genomics England, has applied these pan-tumor to whole populations of patients. And if you look at these mutational signatures, which are quite beautiful to the trained eye, you can see that one second to the right in melanoma, which shows a very, very clean mutational signature profile, which shows that most of the damage that drives melanoma is caused by UV light. Compare that to colorectal cancer on the right, where you can see these two clear subgroups where aging is a driver for about 85% of patients, but there's this little subgroup on the right where microsatellite instability and mismatch repair is the driver, and those patients are the group that would respond to checkpoint inhibitors. So this is very important, how we can apply these machine learning and also statistical techniques to data across our entire data set. So let's bring these last two examples of pathology and molecular biology together into one integrated type of analysis called radiogenomics, which Genomics England are very excited about and we're launching this year. So what we're doing this year is we're adding radiology, but also importantly, digital pathology images to all of our solid tumor patients. And why would we do that? Well, that allows us to explore in one tensor the spatial context and also the molecular context of a tumor in a multimodal analysis. We know that's important. So take colorectal cancer, for example. Colorectal cancer, in some cases, responds well to this class of drug I've mentioned already called an immune checkpoint inhibitor. And what an immune checkpoint inhibitor does is it disables a kind of camouflage cloak that some colorectal tumors represent to hide themselves from the immune system. And what we hoped at first was that by uncloaking these tumors and lighting them up, the immune system would recognize them and the local lymphocytes would attack the immune system. But it turns out that that didn't always happen. And the reason it didn't always happen was because there are some tumors that are embedded in such dense microenvironments that the lymphocytes actually can't see the tumors even if they are uncloaked. So really, response to treatment and prognosis from those patients is very much driven by this combined spatial and molecular context. And so integrating in multimodal analysis these two modalities alongside the clinical data, the outcome data that we also have on our research environment, we think is going to drive really new discoveries about response to treatment, about prognosis, and about the mechanism of action of disease. So let's move on to talk about some of the infrastructure required to deliver these biological results. So in a room full of data scientists and infrastructure experts, let's pause for a moment and consider what remarkable infrastructure DNA is itself. So if we were to do a quick back of the envelope model, 10 DNA base pairs are about 3.5 nanometers long with a diameter of about 2 nanometers. So if each base pair represents a bit, which is probably undercooking it, then 10 bits would be equivalent of 6.8 nanometers square. That's an information density of about 1.5 terabits per millimeter square, and that's about 800 times more dense than the densest hard drive. So we need to hold in vast respect the infrastructure that nature has created for us over billions of years of evolution. Now, despite the purity and the clarity of the signal from the DNA itself, measuring that signal is unfortunately a very, very noisy process. We're measuring individual chemical and physical changes from one molecule at a time on a phosphate backbone and at a nanometer scale. There are 3.2 billion base pairs, 6.4 billion bases in the whole genome. And to sequence this, we have to do this in massive parallel. And so thousands of genomes are processed in parallel on a flow cell. Now, the good thing is that we all know in this room that machine learning is an ideal technology for separating signal from noise. And that's where it comes in genomic analysis. So I'm going to give you a couple of examples. I mentioned before that we're using nanopore sequencing from Oxford Nanopore to analyze our solid tumor genomes. Now, first of all, why are we doing this? Well, it turns out that there's about 8% of the genome which is just simply inaccessible from traditional methods, sequencing by synthesis, which is what Illumina uses. In particular, at the very center of the chromosome, the centomere just can't be read by traditional methods. So there's a lot more DNA out there to read. Also, there are complex variants, which are signatures of cancer, like structural rearrangements and fusions and large translocations, which are very, very difficult to read with short read technology. And lastly, there are these chemical modifications to DNA, which we call methylation, that not only drive disease, but can really affect prognosis and disease classification. So it's really important that we can learn about these additional features. And Oxford Nanopore have come up with a way of doing this. And it's represented by this quite remarkable cartoon on the left. So the way Oxford Nanopore works is that they pull a DNA sequence through a nanopore. It's actually a biological pore. And that pore has a current that runs across it. Now, at the natural pace, that DNA strand would run through the pore at about 1 million bases per second. And so Nanopore have had to come up with a molecular brake to slow that down. And by slowing it down and regulating the speed to just the right amount, they're able to look at the output of an oscilloscope and can identify from that oscilloscope read the individual A's, C's, G's and T's, and also the modification to those bases through methylation in real time as the DNA strand goes through the pore. It's absolutely remarkable that this can actually happen in practice, and not just one pore at a time, but thousands and thousands of pores together. But the amount of data generated is absolutely enormous. 3 billion base pairs at about 340 kilobit per pair would generate 1 terabit for a 10 coverage genome. And actually, Genomics England, we sequence our genomes, certainly the somatic genome, at 70 to 100 times coverage. So that would give us perhaps 10 times that, 10 terabytes per patient. So this is an absolutely enormous file size, and we'll be limited from a transport and a storage perspective from transporting all of that data off the machine. In fact, the sequencing is actually done at a faster pace than we can extract the data from the machine. So some of that analysis and encoding has to be done on the machine itself. And our Oxford Nanopore today uses GPUs actually built into the machine to do about 25% of the base calling. That is the reading of the ACs, the Gs and the Ts straight off the genome itself. So reading bases off a genome in real time is actually a challenge, which is quite similar to speech recognition. And amazingly, Oxford Nanopore employed QuartzNet, which is a recurrent neural network that was designed for speech recognition and is now used to read molecular bases, which is quite a fascinating transfer of knowledge. Why is it similar to speech recognition? Well, it's because sequence matters. Just like in speech, you need to listen to the individual words in order, and in fact, the sound and the enunciation of each syllable affects the syllable after it, and that happens in the genome as well. Reading a C will influence the read of the G or the A or the T that follows it. Now, improvements to base calling have continued at an incredibly rapid pace in the last 10 years or so. And that's been done partly through improving the chemistry of the pores, something that I'm not going to talk about at all, but also partly through improving the algorithms for reading these sequential patterns of bases as they've run through the pores. And that, as I mentioned before, is done partly on GPUs on the Oxford Nanopore devices, but also partly in our data center on GPUs, which are up in the cloud. So that's very much our business, and it's something at Genomics England which is very, very important, because we need to get read quality to the high 99% or even above to be clinically useful. So what's interesting is that that base calling was initially done with a hidden Markov model, which is just a traditional statistical approach for analyzing sequential data. That was quickly replaced with an RNN, QuartzNet, and what really helped was adding transducers to QuartzNet, which improved the ability to make incremental predictions of successive bases without rerunning the entire neural network. We then started analyzing reads in raw format, which meant that the electrical signal could be looked at unsupervised without any labeling or annotation, and that really sped things up. And then finally, we quite recently introduced a flip-flop modality to the base reading, which allowed the neural network to flip into a different mode when it was reading regions of the genome called a homopolymer, which are very long repetitive regions, which any machine or indeed any human would find very, very difficult to read itself, and then flip back when it got to a kind of more normal region of the genome. And all of those improvements together have made successive improvements which now make us confident that Oxford Nanopore base calling is of a sufficient read quality to be tested out in the clinic, and that's one of the projects we're working on this year. So far we've talked about base calling, which is measuring the A's, the C's, the G's and the T's that are native to the human genome, but it's really in variant calling that we find the signatures of disease. So what is a variant? A variant is the way that a human genome varies from the reference genome. And the great news is that all of us are 99.8% similar. We only vary from each other with about 0.2% of our genome. But that 0.2% still creates a lot of variance. If you run the maths, it's about 4 to 5 million variants per human genome. Now, of those 4 to 5 million, most of them are not pathogenic. They create their polymorphisms that create features like blonde hair and blue eyes. But within those 4 to 5 million variants are sadly the drivers of severe disease like cancer and rare disease, and also polygenic drivers of common disease. Now at Genomics England, we sequence the DNA of patients with cancer and rare disease, and we're determining genomic variants crucial for the diagnosis and for therapeutic decision making. Traditionally, the way that we've analyzed the veracity of those variants is by applying a visual genome browser to the genome. And we make that available to clinical scientists in the NHS, and we've got a picture of that genome browser on the screen on the left today. So it's a little bit hard to see, but if you look closely, you'll see that we, in this case, are interrogating a gene called TP53, which is actually the most prevalently mutated gene in cancer today. And all of the horizontal grey bars are the different reads in which we've read that genome repeatedly. And the reason we have to read it repeatedly in cancer between 70 and 100 times is because there's so much noise that we need to get high statistical confidence that we've read those variants accurately, otherwise we may misdiagnose that patient. So this is a challenge. It's a challenge because it's difficult to get humans to look at every single one of these pile-up images and assign probabilities. But it's a challenge as well because there are very many types of read issues which drive inaccurate reads of variants. And I'll explain them here. So deep learning has turned this challenge into a computer vision problem. There are 6 features of quality per variant, which are now measured as standard today, and I've listed some of those on the left. There's the individual base quality, there's the noise from adjacent bases, there's the comparison against the reference genome. Some parts of the reference genome itself have higher inherent quality than others. And all of those features need to be taken into account and are taken into account by highly trained clinical scientists today when they're manually reading those genomes. But that's just a process that doesn't scale. Now, we've been working with Google, who have come up with a very impressive variant caller for long reads, which is called Pepper Margin Deep Variant. And what Google have done is that they've taken those 6 features and they've assigned them to 6 pixels. And for each candidate variant, these 6 features are encoded into a pixel to form a visual representation of the region. And we can take those visualized maps with the variants encoded and compare that to the truth set, which is generated by an organization called Genome in a Bottle. And by training through classical image classification, we can actually use deep variant to classify which variants are true and which are false. And that's the basis of a variant call file, which travels into our clinical interpretation engines and drives decision support on whether it is indeed a disease, whether there's a licensed drug for that disease, or whether there's a clinical trial available for that patient. So it is remarkable how, first of all, deep learning and also how Google are contributing to a massive analytical challenge of calling variants from a genome. Now, GPUs are essential for accelerating that variant calling. If we were to do that exercise with a CPU, you can see it would take about 5 hours to run the genome of a single patient. With GPUs, it's down to about 12 minutes. And you might think that that's not so important, but in fact, this is deeply important in the clinic. So we are sequencing children with relapse cancers where doctors need to make urgent treatment decisions, and this data is a key input into those treatment decisions. So getting the turnaround time from the sequencing back through the genomic lab hubs, back to the clinicians, quickly and efficiently, and of course importantly accurately, is absolutely mandatory to clinical adoption of this technology and to the well-being of our participants, and something we take very seriously. And I think the work that NVIDIA is doing by investing in GPUs at the edge of these boxes, but also in our data center, is central to this effort. It's hard to understate the importance of this computational improvement. So in summary, machine learning architecture is absolutely essential for genomics at scale. The scale at which we're producing genomic data and the way that this needs to be processed both in real time and in post-sequencing in order to be clinically useful and useful to scientists means that the only viable way of doing this is through having appropriate and hugely scaled machine learning architecture. So we're delighted that NVIDIA has invested in over 100 exaflops of GPU capacity in the public cloud in the last 12 months. We're heavy users of the public cloud with Amazon. We're really hoping that some of that investment has gone into the London instance, and we plan to be putting your GPUs through its paces in the coming years. So I've covered a lot of ground today. It's very clear to me that NVIDIA is transforming machine learning, that machine learning is transforming genomics, and that Genomics England, in partnership with the NHS, is transforming cancer and rare disease outcomes. It couldn't be a more exciting time to be at the confluence of these exponentials of biological knowledge and machine learning. We're hiring, please come and join us. And most of all, I really look forward to an active Q&A. Thanks for inviting me, and let's move on to the questions.